You do have to have some tolerance for critters for
Sure.
biological control.
Right, right,
Like we have swallows that make nests all over everything in the spring and the summer, and they're great at keeping down all the flies and everything. But you have to have tolerance for the barn swallows and
right,
them messing up the side of the house where they're making their nest and stuff.
right, right. That's a good reason to have a metal building. You just hose it down,
Yeah,
and you'll be fine. Yeah.
yeah.
Welcome back to the Idea to Impact podcast, where we are talking to faculty, inventors, and entrepreneurs and bringing you their stories of experience with commercialization and technology transfer. This is episode with Dr. Larry Griffing. It's part 2 of his episode. And in today's podcast, we are going to talk more about Dr. Griffing's research, his experience with the ICOR program, and with his Innovation X grant that is helping to teach students not only about the research he's doing with herbicides and biology, but also teaching them about commercialization and how that process works. Let's get started.
And for those that are listening from the beginning, we're talking about various. He contributed to many fields of natural philosophy or science back then. And you're able to discover this, and I think you even published on this portrait. Is that correct? And and then taking that now to what you're currently working on, because I found this also very fascinating, into ways of, I don't know if it's getting rid of weeds or is it making sure that weeds never start, and it's an entirely non-toxic way of doing it, I believe. Is that correct?
Right, right. So
What's the layman's explanation that I probably got wrong there?
no, that's right. So we have different kinds of herbicides out there. We have herbicides that you spray on a plant like glyphosate or Roundup, the plant dies.
Okay.
It's just like
Okay.
it's a really potent herbicide. Unfortunately, a lot of the plants are becoming resistant to it. So that's evolution.
Right. Right.
And the also there's questions about its safety. There's some worry about the safety of glyphosate, or maybe the carrier that glyphosate is in, or something like that. And in fact, both glyphosate and dicamba have been outlawed by the European Union. And so these are important herbicides used to really reduce the number of weeds out there. And both of dicamba is like an analog of a plant hormone, and when you spray it on it, the plant just goes hormone wild and dies. Whereas the glyphosate inhibits the process of photosynthesis, and so it dies because it can't photosynthesize anymore.
So you starve it, the latter one, and the other one just is like a bodybuilder on steroids, and then the
Yeah,
muscles blow up or something like that, or
yeah, it becomes hormonally imbalanced, totally
gotcha.
hormonally, and doesn't know
Okay,
where to grow out anymore and that
gotcha.
kind of stuff.
Okay.
So back in the day, in the 50s, 60s, 70s, 80s, 90s, people have been looking for herbicides, nice, effective herbicides that don't hurt people, and there are a bunch of herbicides out there. An example is are, and they often work at a subcellular level. So the microtubules inside a cell is kind of the cytoskeleton, the skeleton inside a cell.
Hmm.
The microtubules are in plants and they're also in animals, but the microtubules in plants are sensitive to drugs that plant microtubules aren't sensitive to. And so one of the herbicides, for instance, is a microtubule depolymerizing agent. Um, that's one mode of action of herbicide that's out there, and a lot of them target other things that are particularly unique to plants. So one of the things that's particularly unique to plants is their sterol composition. So sterols are all of those kind of fatty components that are in the membranes of cells that help to regulate the function of the cells. And we know about it because if we get cholesterol in our arteries, it builds up a fatty deposit there, and it causes atherosclerosis, so the blockage of the blood vessels. And so if we're not able to take up the cholesterol that we eat, then we get hardening of the arteries. And so if you look at the cereal boxes in your pantry, you'll see that it says, "Oh, no cholesterol," in there. It's not true. There's just a little bit of cholesterol in plants, and there's low enough concentration so that the FDA can let them say that there's no cholesterol in there. But there's other sterols that are unique to plants. And so people for a long time, ever since the 60s or so, have been interested in thinking there's a way to inhibit the synthesis of sterols in plants so that they don't grow as an herbicide. And they found a whole bunch of chemicals that inhibit the synthesis of sterols, and they all have a disastrous effect on plant growth, but they also have a disastrous effect, or in some cases, a beneficial effect on animals in very low concentrations. So statins, which are taken to lower your cholesterol, were originally identified as an herbicide.
Oh, interesting.
Yeah. So lovastatin is an effective herbicide, but it also affects our cholesterol metabolism. We don't want a whole bunch of statins out there. It's also really expensive. So what you want is something that's really low expense that can specifically address that single important growth component of plants. And so what we discovered was we were interested in how sterols get into plants originally. And we were looking at membrane contact sites, the contact sites between different organelles that make them all talk to each other. This is a new area. And it's really exciting because people often thought that the mitochondria did mitochondria things, and the chloroplast did a chloroplast thing, and the peroxisome did a peroxisome thing, and the ER did an ER thing. And it was all separate. But it turns out that it's all connected. Not surprising, but there you are. And so we found that the connection between the plasma membrane and this membrane system inside the cell called the endoplasmic reticulum is a place where sterols can get out from outside the cell to inside the cell. But in order to do those experiments, we had to figure out a way to deliver the sterol to the plant. And people in the past had been doing that just by dissolving the sterol in ethanol and then adding it with a lot of stirring to a solution. But we found that doesn't get the sterol in. That just doesn't work. And so when we discovered how to get sterols in, we thought, oh, we could also solve another important problem that people have had in this area of science, which is people realized that if you had mutations in the biosynthetic sequence leading to sterols, you got really severely dwarfed and non-germinating plants. Okay. Now, if that's truly just in that biosynthetic pathway of just the sterols, presumably you could add back the sterols to it, and it should make it be normal, right? So a test of whether you're getting the sterols into the plant in this case would be whether or not you can chemically reverse these mutations, and we found that we could. Wow. But the other side of that, we did this all in just one experiment, one petri dish. It was great. We had a mutant growing, actually not growing. We added a little bit of sterol to it in our special solubilization agent to get it in, and it completely reversed the growth. It looked normal.
Wow.
But then as we increased the sterol concentration, the mutation looked like it came back. Huh. It was so weird. And so I didn't know what was going on there. And what we found is that when we added these sterols to plants, all of these symptoms of mutations in the pathway appeared. And it turned out when we actually measured the sterols inside the plant, that by adding this higher concentration of sterols outside the plant, it cut off all of the biosynthesis of the sterols by the plant. It was just like causing mutation in the pathway. So there is a feedback inhibition
Mm.
by these sterols on the growth of plants or on the production of sterols. So even though we were adding a whole bunch of sterols outside, their sterol concentration was actually going down, and that's the basis for this discovery. And so it's a very basic biological discovery, but it has this amazing potential influence on how it could help people. And that is that the sterols, these plant sterols, are often used in gram quantities for people to lower their own cholesterol. And yet you can, and so you could put it on the soils, and it would break down, and it would be completely non-toxic.
So, as a researcher, I tend to think, okay, you've discovered this new, we'll call it technology, this new herbicide. I used to think that that's where it stops. Like, okay, we've got this great discovery, you publish a paper on it, you get recognized for it. But then there is this whole other side of it, especially when you're working at a university, because the university, um, technically owns this discovery. So then you take it further. Now you're trying to commercialize it, right? You're trying to do something, not just have it be this great discovery, but now you're trying to get it out into the world and let it have some impact, right?
Right. And so actually, this was kind of a side project. I wasn't getting funding from any place other than just the biology department on this. So we didn't have like National Science Foundation funding for it or USDA funding or any of these kinds of things, which is kind of rare.
Mm.
But it was completely university funding that provided us with a material that we needed to make the discovery. And so I made a invention disclosure to the technology group there, and then they looked at it, and we talked about whether we should patent it, and we started looking at prior art, as they say, of whether these discoveries had been patented before, and we didn't find anything. And so a year ago, I made a provisional patent to the U.S. patent group,
Office.
and um, then we extended that just this year to an international provisional patent.
Mm.
So we have it protected to in the sense that we have a while to prove its effect while we have it as provisional. So we're now doing field tests on it to see what we can do to establish its effectiveness out there in the field.
So we talked about two different uses of your discovery. One was as an herbicide, which folks in the agricultural industries would be very interested in. And then there was also, you talked about people taking it in gram quantities to reduce their own cholesterol. Is this the same thing we're talking about for two different uses, this patent? Or are we
Yeah.
doing one?
So we didn't patent anything that had to do with nutrient supplements.
Gotcha.
One of the problems is that uh, that's already been nutrient supplements is kind of old news. So you can go down to the local store, to the local nutrient supplement place, and buy plant sterols and take those. The problem with it is that again, um, and they even use some of the same mix that we have in order to get sterols in, but it's fairly unregulated. It's fairly cheap,
Mm,
and so it's not a drug. It doesn't have the same kind of drug
right.
uh, requirements that are necessary for. But that's the other nice thing about this discovery is that because it's non-toxic and not really a drug, we don't have to get it uh, analyzed and approved by the
Mm.
Environmental Protection Agency. So the EPA step is a huge step, a huge hurdle
Oh
that
yeah.
normal herbicides have to overcome, and we don't have to do that.
There's so many, alphabet agency regulations that we've talked to other people about from, definitely the FDA, EPA, that we're taking them more than a
Years.
decade, right? I mean, a dozen years or more, just before they could start really looking at commercial applications. They were still in trials, so that's a huge advantage. And if I'm in
Mhm.
this, if I'm in the ag industry, let's say I grow cotton, because cotton is a big thing around here
Yeah.
in politician area. Um, why? What benefits would your technology give me that maybe I wasn't already getting, or whether it's a direct benefit or an indirect benefit? Because I get the
Oh,
non-toxicity of it.
yeah. So it would be a direct benefit because for we haven't tested it with lots of cotton plants yet, but
Right?
conceivably it would actually be cheaper for them to use this to keep the weeds down before they plant cotton
Okay.
than the current herbicides they use. So they currently use pre-emergent herbicides, but they're kind of expensive. And what we're trying to do right now is make a formulation of it so that it lasts a long time. They don't want to go out and spray their crops over the winter several
Right.
times in order to keep the
Right.
weeds
Right.
down. The weeds don't actually grow that much during the winter, but they don't want to do a lot of spraying in the winter, of course. And so we're trying to figure out the formulation that would be best for that. And that's probably the biggest challenge we have right now is working on the formulation. But getting back to the cotton farmer, we figure we could probably get it down to $10 an acre.
Hmm.
And they're
Wow.
currently paying like on the order of $150, $200 an acre for their
Oh, wow.
herbicides. So
It's a
we have a
whole order of
considerable
magnitude
cheap,
less. Yeah.
yeah, it's a lot cheaper. So glyphosate is $5 an acre. But the problem with glyphosate these days is that it isn't the glyphosate that costs the cotton farmer money.
Right.
What costs the cotton farmer money is the glyphosate resistant seed that Bayer sells now. That's really expensive.
Right. Right.
And so that's actually one of the nice things about this. Using an endogenous chemical that's already in the plant and just having it feed back on the biosynthetic pathways for these, it's probably going to be difficult for them to acquire resistance to it.
Interesting.
So there probably will ultimately be some resistance to it, but it might just require more or a slightly different sterol or something like that. But since it's such an integral part of their regulation
Right.
of growth, just adding this sterol to inhibit the whole sterol biosynthetic sequence might be hard to overcome. What we're working on in the lab now is, like I said, we were interested in sterol transport. One way that they could become resistant to it is if they blocked the sterol from getting into the cell, from them being able to sense it. And so we need to figure out what the transport system is and how easy that would be for the plant to change.
Okay.
And going
That
back to kind of the kindergarten version of this, if you're putting it into the soil, it's not going to negatively affect the cotton, right? The cotton's still going to grow well.
It will affect, actually it affects small seeded things the most, which are mostly weeds.
Okay.
So it affects all these small seed. The bigger seeded things are a little more resistant to it because they probably have a little bit more sterol in their seed to rely upon when they germinate.
Okay.
So cotton has a big seed, corn has a big seed, beans have a big seed. So all of those things are a little more resistant to it. But the main thing is that because it's a natural product, it's almost, the way I view it is it's almost like the active ingredient in mulch. You know, when you add mulch to a garden, things don't grow.
Right.
Yeah. And it isn't just because it's covering up these little seeds and they can't poke their way out. There's an, inhib- an active inhibition going on there.
Hmm.
And this sterol might be part of that. I mean, that hasn't been proved yet, but it would be easy for it to be that. And so consequently, we think that this sterol, when it gets into the soil, is broken down fairly rapidly. So mulch only works for a short time, and why is because the active ingredients gets broken down. So the arthropods in the soil, the little bugs,
Right.
tend to eat plant sterols and break them down, and that kind of thing. So we're also interested. One of the main things that we're asking money for now through granting agencies and whatnot is doing a chemical analysis of the soils to look at the rate of breakdowns of these sterols to see how long they last in the soil. So getting back to the cotton question, if you put it on, this is true for all pre-emergent herbicides. You put it on, they last for a while before they get washed away or broken down or bound to the clay particles or whatever it happens to them, and then you can go in and plant your crop again.
I see.
There's a certain time, and so we're looking at that time window right now when our herbicide would be best relating to when you plant the cotton. And then the good thing about this herbicide that we have, is that once the plant gets about that big, once it gets like a couple inches tall, you can add the herbicide, you can add the sterol, and it has no effect on plant growth.
I see. Okay.
So you could wait for your cotton to grow a little ways. And actually, cotton and a lot of the other plants, since this is completely organic, one of the major markets for this would be in cannabis production. Organic crops, vineyards, trees, but the emergent crop for this is cannabis. And once you get a cannabis canopy, where the leaves are shading the
Right.
background, they don't grow weeds. There's not a weed problem. And so what you want to do is be able to inhibit the weeds after they grow a little ways. And then you could put this on the plant. It would keep the weeds from growing before the plant got big enough to actually shade itself, so that it didn't have any more weeds.
There's an old parable about the wheat and the tares, or the wheat and the weeds growing up together, but you can actually now make it so that the weeds don't grow with the so to speak, right? That's pretty cool.
Which could be really important now with this last election cycle. A lot of other states have legalized marijuana, and so that's a whole other podcast topic, is just how much that industry is growing and the opportunities that are going to come out of the legalization.
Right.
Yeah, in organic crops, it kind of depends on the level of mechanization they have out there, but people will spend $300 to $1,000 an acre just getting rid of their weeds.
Wow.
And as I mentioned, we can probably manufacture this thing at $10 an acre. And so we'll probably price it a little higher because the value to the farmer will be a little bit more.
Absolutely.
But the possibility of making a profit on this for Texas A&M and for us, the company that I made is Griffin Biologics, is pretty good. The profit margin will probably be pretty good.
So speaking of that, it sounds like, from what I read, you did an ICOR project to get Griffin Biology started, to get this technology, to get some customer feedback. Talk about the ICOR process for you.
Yeah, that was really good. We really enjoyed doing that, and we were really lucky. We had the last ICOR before the lockdowns.
Wow.
So we had it up in Dallas in February.
Okay, this year then.
Yeah. And so we did 150 interviews of people at all levels of the business. That's the cool thing about ICOR, is they tell you to get out the building, don't try to sell your product, but find out if there's any demand for the product out there, because most startups fail because there's no demand for the product.
Right.
And so then there's this question as whether you have a product market fit. Once you identify the markets out there, does your product actually fit that market? And so we actually stumbled upon the organic crop area through the ICOR program. We were thinking it would be more for row crops and that kind of stuff, but there's a big market out there for the organic in the organics area, and that's probably how we are going to make this company scalable, is to start by offering use licenses at use for organics and certain crops, then use for the row crops, or use for turfs. I mean, one of the other things that we found in the ICOR through our interviews was that there'd be a pretty big market for this in the golf course industry and in some places where they just don't want weeds at all to ever appear, like on aircraft runways and things like that. The amount of horrible chemicals they put on aircraft runways is crazy. We were able to, when we were up in Dallas, and we were able to go to DFW, to the guy that was in charge of keeping their runways going, and he said he'd love to have a product like this.
Wow.
Because they were right at the limit, right at the allowable limit for getting the runways cleared.
Wow, that's kind of scary. I fly at a DFW.
There's a lot of runoff. I mean, that's a problem.
Yeah, it all washes down into somebody's water somewhere, right?
Yeah.
Another reason why I won't ever live in Dallas.
Well, I think he was a really good guy, and I think they did it safely. I did it within the EPA limits.
Yeah.
That's actually one of the things that we were surprised by when we did the ICOR interviews, was that a row cropper, a guy in the Brazos Valley, is not interested in the safety or the organic component so much, the non-toxicity. All they have to do is follow the directions on the package. If they follow all their directions, the EPA has already done all that work for them to figure out if it's going to be safe or not. So they have to have their own personal protection equipment and all that kind of stuff to mix the herbicides and all that kind of stuff. If they follow all the instructions, they don't care about the toxicity of the product, because the toxicity has already been controlled for through EPA regulations. And OSHA.
That's a great discovery right there. It's something that you wouldn't necessarily intuit, but it makes total sense once you discover it, right?
And how easy was that process for you? I mean, you're a scientist. You kind of used to being in the lab. Was it easy for you to go out and talk to people, or was there like a learning curve? I mean, was it out of your comfort zone?
Well, we had a couple people on the team, and I was kind of comfortable with it because I can talk a lot, and and a lot of this thing is you kind of got to keep your foot in the door while you talk to people about things that matter to them.
Right.
You get the conversation going, and you can't really be shy about it too much. And so there were people on the team that were more shy, and I think they got much better at it over the time of the interviewing process. And so it's about 6 weeks, and so with 150 interviews, we were able to get lots of good practice talking to people about this kind of thing, and we learned a lot about the whole industry, which was really important. And even with learning all of that, I mean, as just being able to get an organic herbicide registered just varies from state to state. So there's all these regulations that are associated with organics that are different from other herbicides that are managed by the EPA. So once it's listed by the USDA, California has a big organic herbicide certification system, and it varies over state to state to state. So we would like to partner with some organic product distributors who know all of these things, and then license it to them so that they already have a customer base that they can go to. We'll probably never be in the business of mixing this herbicide and selling it to the farmer. We'll probably give it to people, give the intellectual property to people so that they would be able to do that and make a big profit off of it.
To keep focusing on the research and to continuing with developing and making new discoveries while somebody else takes the lead on getting it used and out there in the market. Yeah, that makes sense. It's a different way of doing business,
Yeah, well, the organic distributors either have or they know people that already have the stuff to mix it in and how to do all that stuff. So that's a really efficient way of doing it. And is Griffin Biologics looking to do things beyond what you're currently working on in the herbicide market, or is this?
Well, so we keep discovering new things about this, and so we're just in the process of putting together another disclosure
Oh, really?
that relates to this. Yeah. So yeah, it will keep going. I think that as we realize more of the mechanics of how, for instance, sterols get around plants and what they're important in terms of germination or growth of the seedling or even flowering or some of these other things. I think there's other patents that we could make that will potentially be useful for people out there. But again, we probably don't want to be the manufacturer per se. We just
Right,
want to
right.
be able to share the intellectual property with other people or sell the intellectual property to other people.
Mhm. Mhm. So you did the ICOR and then you applied for the Innovation X grant, correct?
Right. So the Innovation X grant we've extended some of the experience that we had with ICOR to undergraduate and some graduate students. And the idea behind this was originally the students would actually get out in the field and work with us a little bit, but it's fall and we have this kind of lockdown mentality right now where it's hard to get people out there.
Right.
So what we decided to do was for the first semester, we took this kind of novel approach where wanted the students to get the same feeling that we had while we were doing the ICOR. And that was they had to have a product, but they didn't know what the market was for that product. And so the product that we decided that they had was the skills they get at Texas A&M,
Hmm.
And so now the question is, are there companies out there that want to hire them
Right,
because they have that value? So they have to go around and ask what the value propositions are out there, what their value is to potential companies. And it's been really interesting. We have chemical engineers, we have biologists, we have planty people, marine biology people, all different kids at various levels in
right,
it, and they're all interested in getting a job when all
right,
this is done. And so we're trying to get them to go to the second level, where you go to the HR people initially and they say, oh, we want a well-rounded person that can
right,
communicate well. And of course, that's what everyone says. But who, where's the edge? What do people really want? What is it? And you can usually only get that when you get past the HR people, where you're kind of communicating with those, and they're learning about that. And so they've had a lot of interesting experiences so far that are very, very similar to the ICOR experience, and we can use that analogy of their product,
yeah,
and they're essentially making their own business of them
right.
in this startup
And the
mode because
customer
they're
and
all
client
starting up.
discovery. Yeah, they're discovering what their clients want. So are some students switching majors based on what they're finding?
I don't know.
Like, oh, no jobs there.
Yeah.
Yeah, yeah. Well, actually, I think they're refocusing their undergraduate curriculum a little
Right,
bit. Maybe not switching majors completely,
right,
but knowing what better courses to take and
right. To pick up at least
internships.
those ancillary skills that would allow them to play a little bit toward that market, whatever it might be. I
Yep.
think that's wonderful. I think it's a great. It's not only a cool application of the process, but from what I've seen from the time that I went to school, which was right on the cusp of getting an education for education's sake was valuable, and it's really just a high level trade school. You know that you're trying to get a job with this. I was right in the on the cusp of that. That's I think fully flipped over. That the value proposition of most higher education institutions right now is jobs. Right there are outcomes, uh, and so looking realistically at what those outcomes might be as opposed to the pipe dream that you have in your head or that you maybe are selling your parents on. That's incredibly valuable. I love that idea.
Well, and it also gets people to be more independent because a lot of the people are just doing what their parents told them.
True.
Yeah,
Right.
yeah. Oh, go be a doctor, go be a lawyer. You'll make lots of money, and the
Right.
parents probably don't know anything about the field, but they know that who's ever a doctor or a lawyer probably gets adequate money to do what they're doing. But And so, I see a lot of people like that in biology, where they're majoring in biology because then they can apply to medical school. But the trick is that medical schools accept people from all over the place, not just biology. And so it's kind of uninformed. It's an uninformed approach to getting what you want out of life. So that's the other thing I assigned to the students was a psychology course on which is through Coursera.
Right.
We're not supervising this at all. I just kind of gave it to them as a background information, but it's called The Science of Well-Being, and it's a really cool course by this psychologist at Yale who talks about what is it that makes people happy. And when I'm telling them that they're going out to try to find a job, what is it about the job that's going to make you happy? Is it more money? And the answer is probably no. And
Right.
what is it about it that actually makes you happy? And actually, one of the things that I find completely gratifying about this whole commercialization aspect is um, I can see a way that my research helps people, and that makes me happy.
Is that Paul Bloom at Yale who's doing that?
No, it's a lady. I forget her
Okay.
name. Yeah, but I can send you the link.
Yeah.
I really like that. I think we've heard that from a lot of inventors that we've had on the podcast about, they love their research, they love what they do, but what really makes them happy, what really keeps them going is knowing that they're doing something that's having a positive impact on the world. And I mean, certainly, it sounds like what you're doing is important. I mean, it's important for people to have plants that aren't going to kill them because there's some sort of chemical. I mean,
Yeah. Well, and then we don't kill everything else. I mean,
Right.
some of what we do actually is so interesting. We're dealing with this chemical in a plant, but it turns out that that chemical is not made by insects, and insects require it. Insects can't make cholesterol.
Ah. You
And
see, I
and,
thought
and
when all the insects were eating all the sterols, I thought they had a cholesterol problem. They were just trying to lower their own cholesterol. But not,
they need the sterols, and
gotcha.
so they go in and they eat
Oh,
plant sterols in order to make the molting hormone.
interesting.
And if they don't get the sterols from the plant, then they don't molt and they'll never get to the adult stage. And so that's actually one of the future patent applications that we're looking at, is how to use this as an insecticide, basically.
Right. Yeah.
At the same
In the
time.
same way, just not letting them get big enough, right? Yeah.
Right.
That's fascinating.
Yeah. There's other ways. Conceivably, we could make some plants more like the weeds, more tasty, and have the crops
Right.
less tasty.
Right.
And then you wouldn't have to go around killing all these bugs. You could just have them eat your enemies.
Yeah.
Right.
Well, I can see that being like an insecticide or pesticide or whatever that would be good. Like indoors, people have like pets that they want to protect their plants they have inside, but they don't want to do harm to their pets if they happen to eat the plant that you sprayed it on. But it would help with things like fungus gnats and other insects that are growing on plants.
Oh yeah, well, people can't use malathion, for instance, in any places where there's fish, and yet it is one of the more popular insecticides around.
Mm-hmm.
That would explain how my fish keep dying. But
well, you also don't want to use the public water for. we have horse troughs out on our farm, and they have a well. They use well water, and because they're great big horse troughs, they easily generate lots of mosquitoes.
Right.
And so we put little bitty feeder fish, little bitty goldfish in there, and they get like this big.
Oh wow.
Yeah, you go on PetSmart and you get the little bitty goldfish. They get really big and they're really pretty.
We got a mosquito
and they cost 65 cents. But if you use city water, they all die.
Well, it's chlorinated to an almost impotable level, isn't it? The
Yep,
chlorine levels are
because
that
of the
high.
Here
chlorine levels.
we
Yeah.
are talking about how bad College Station water is, and I'm still sitting here like I like it.
Yeah, Beth's from College Station, and so she grew up on this. But I got to tell you, if my wife takes a shower and like I give her a hug or something, and she smells like she was in the pool.
Yeah,
The chlorine that's not just on your skin. And my one
Mm.
son, the one that kind of finished high school early as well, he looked all into it and he said, "I think we're like 0.01, Dad, away from the EPA saying we have impotable water." Like, wow. So that's good to know. What I like to know, I was wondering too, on large land, if there was a mosquito problem, my my solution was going to be to get rid of all of the water sources. But I love your idea better, which is use the water sources almost as a trap and let the fish eat them. Right?
yeah. Oh,
Yeah,
and it works great.
yeah.
Yeah.
I'm
So
definitely
I often find that adding things that poisons and things like that to get rid of pests can work so much against you. You
right.
know, there, yeah, you do have to have some tolerance for critters for
Sure.
biological control.
Right, right,
Like we have swallows that make nests all over everything in the summer, and they're great at keeping down all the flies and everything. But you have to
right,
have tolerance for the barn swallows and
right,
them messing up the side of the house where they're making their nest and stuff.
right, right. That's a good reason to have a metal building. You just hose it down,
Yeah,
and you'll be fine. Yeah.
yeah.
That's, so you mentioned a book that you either just finished or are about to finish. You said one of your colleagues was reading it on imaging. Is that correct?
Yes,
Talk
yes.
talk a little
It's
bit about that.
yeah, it's called Imaging Life, Photography, Microscopy, Biomedical Imaging, and Image Analysis. So it includes all those things.
Right.
What was the first part of the title? I kind of got you cut off a little bit.
It's called Imaging Life, and the first part of the title, the subtitle is I cover photography, microscopy, biomedical imaging, and image analysis. So it's actually uh, written from a completely kind of applied digital point of view. So when I was learning how to do imaging, we were using film. We there was a
Right.
completely different way of taking pictures than we do now. But this, I mentioned film in the introductory chapter as an old technology that is interesting historically. But other than that, the whole book is about digital imaging, and I cover different kinds of cameras, different kinds of ways of acquiring images. I have a chapter on the eye, that kind of stuff. But then I also have about a third of the book on image processing and analysis. And then the last third of the book is on more sophisticated modalities. So one of the problems that I see is that people, for instance, when they're graduate from biology here, they often don't know biomedical imaging. It's usually the biomedical engineers that actually teach biomedical imaging, like MRIs or CT scans or ultrasound. And so I have all of that covered in the book too, where they actually learn that. And I'm trying to do it all in one voice so that you're not having to go to one website and another website and another website to try to do it, and have it interconnected so that I can say, well, back in chapter 6 you learned about this, and here we're applying it here in chapter 13. So it actually makes a cohesive unit by which people can learn about imaging.
So besides doing like a full color Mandelbrot set and like putting it up on your wall or doing like a CT scan or something like that for medical purposes, what would imaging like how would we be able to use imaging in biology and in the sciences?
Well, so actually almost a lot of research is almost completely imaging based these days. So,
Okay.
for instance, um, as I was talking about cryo-electron microscopy, is able to see single atoms now.
Right.
So you're actually able to purify a protein, put it under an electron microscope, and actually see the individual atoms in that protein.
Wow.
Yeah, it's pretty amazing. And so I talk about that a little bit. But the other thing that is very cool is that we think of CT scanning, this X-ray scanning that we
Yeah.
have for coronavirus and all that kind of stuff. There's been some amazing recent work in that. And one of the other X innovation projects is Heather Prestridge's project, where she's looking at micro CT scanning of her whole biodiversity collection. So they have a biodiversity collection of all these stuffed animals and all these things in jars. You go into that area, and it's kind of like the room of prophecy in Harry Potter, where you're walking down these
I've walked into there before, and you've got some strange creatures in there. Yeah.
But they take these little fish or whatever, little crabs or turtles, and they put them into a CT scanner. And you have big CT scanners, but for the little fish, you use these micro CT scanners, and they are being used all the time now for important 3D reconstruction of things. So they're pretty good resolution. They have fairly high resolution, but if you're trying to look at all the connections and nerves in the head of a fruit fly, you want to when you take that sample out and you're going to section it, physically section it for electron microscopy, you want to make sure that there's no flaws in that head that you
Right.
have fixed. And so micro CT is great for that. And so neurobiologists that are now learning about connectomes of things use micro CT a lot, but they're also used for biodiversity, where now you can actually look at the bone structure without dissecting the fish of all of these different fishes. And so Kevin Conway, who works over in wildlife and fisheries, he looks at all the different speciation going on, and you can actually get really interesting differences between different species of fish that's only really apparent on the basis of their bone structure.
That's
Wow.
cool.
Wow. And you said a lot of students don't know how to do this kind of imaging. You said you teach a lab that teaches your students, and we were talking about yesterday how all this has uh, you've been able to move it over with we've been in COVID and labs really haven't been open. So talk a little bit about how you've switched your lab to online or you know hybrid or whatever.
Right. So I'll be teaching hopefully the imaging class next spring as kind of a hybrid thing, where we have lab pairs, and one of the two pairs will be there every other week.
Mhm.
And I think that actually is good because they can Zoom with each other while
Yeah.
they're doing it, and that actually puts different people in the driver's seat every other week. And I find that when you have a lab pair, there's usually one person that's the note taker and one person that's the doer.
Mhm.
Excuse me. But and so I don't. This gets them out of that situation. So it's actually kind of good that way. I mean, some of these distance learning components are good. So I actually have a lab in advanced cell biology this semester that we're doing completely online. And so what we had to do was reimagine it so that we could give them a good um, uh, theoretical and hopefully practical knowledge of how to do laboratory work in cell biology. And so we expanded the course actually because we knew that they weren't going to be coming into lab and doing the labs. We expanded the course to include a very heavy component about experimental design. How do you actually design a good experiment so that you have really robust results at the end? How do you validate all the components of your experiment? And how do you ask the question so that you aren't unintentionally biasing your own work?
Right.
What happens all the time is if you're doing hypothesis falsification, you win if you falsify your hypothesis. But on the other hand, no one will give you money if you have negative results. And
Mm.
so consequently, you need to have
Right.
a model building kind of approach. And this book that we have by David Glass on it really does a nice job of setting up these different experimental models and how they are addressed by asking questions rather than just stating hypotheses. You still need to know hypotheses. You need how to convert your hypothesis into a question, but and vice versa. But it's really more of an open-ended questioning thing where you are learning about the system rather than just disproving your hypotheses as you go along, falsifying your hypotheses as you're going along. And the kids have liked that. I think they've enjoyed that part. And we moved them through with guided inquiry on all the different organelles inside a plant cell and how they work with each other and how they move. The cool thing about plant cells is that, you know, the plants don't really move very fast on the outside. But if you look with a microscope on the inside, and you might have seen this in introductory biology or something like that, you have this cytoplasmic streaming going on. And it's kind of hard to see if in a regular plant cell. Uh, you have to be able to see the individual organelles pretty clearly in order to actually see all the streaming that's going on. And one of the worst things that happens is that you give people bad equipment, or you give people dead creatures to look at when they're supposed to be alive. And then they look at it and they think they're failures because they can't see what's supposed to be happening there. And so I think we've completely overcome that part where we as we're going along, I can troubleshoot, and so we do it again, and then we fixed it, and so that works. But now, the whole final part of the class is them just building an experimental model and suggesting to us what kind of experiments we should do. So the experimental model is based on a discovery we've made recently in the lab, where if you shine a certain wavelength of light onto this junction between a chloroplast and the endoplasmic reticulum, the plant lights up with a calcium wave. It's actually almost the same kind of calcium wave that you have when you have a sea urchin egg or something and it fertilizes, a sperm penetrates it, it generates a calcium wave through it. And people hadn't seen this calcium wave before. And so we're trying to figure out what the downstream events are in this signal transduction pathway that the calcium is acting as a messenger for. So
that's fascinating.
They're coming up with great hypotheses and models for it, and it's fun.
Wow, that's so cool that that's done online, and listening and hearing about courses like that makes me want to go back to school.
Yeah. I want a professor like you though that really teaches me how to ask these questions as opposed to just learning a linear sequence, because it seems to have helped you in I can see in your thought process even like, let's make the weeds tasty. You're asking questions about what are solutions. So it must be a fun course. Any students that are watching or listening to this, you might want to look into Dr. Griffing's labs or courses on that.
Yeah.
So what's next? What's next for you as far as moving forward, either with Griffing Biologics or for you personally within your own research?
Well, so we're just in the process of applying for a SBIR or STTR grant from NSF.
Okay.
The nice thing about going through the ICOR is that we have established a product market fit, which is a good thing for the SBIR, STTR. We have some kind of track record going into this. And so now I'm kind of learning how to actually build a company. You have to have some kind of company there in order to hire people and make a payroll and all that kind of stuff. So I'm working on that. I'm trying to do it so that I don't have to pay a whole lot of other people to do it right now. So it's a pretty steep learning curve for me. And my assistant director, who is my graduate student and now a postdoc. That's a big part of it. But the other side of it is that I've been gratified by some of the outcomes of some of these courses that I've been teaching. So one of my graduate students took this advanced cell biology course, and she's just really kind of carrying this whole project forward. She's my TA in the class this year, and she's asking cool questions and getting the students to be there's kind of a barrier between the professor and the student, but if you have the TA kind of in between there, that can actually facilitate a lot of discussion and stuff like that. And she's doing great with that.
That's good. Good TA sometimes so defined too.
Plug some SBIR resources. I know they have a ton of resources online, but our office with T's, we're hosting an SBIR workshop in the spring. So it's going to be open to grads, postdocs, and faculty. So, I can send you information about that. It's going to be in the spring, and it'll be 2 hours a week over, I think, 3 or 4 weeks, where we're going to have different topics, all the way. We are going to talk about ICOR, because ICOR is an important component of being ready to apply for an SBR, and it really kind of helps set you apart from other applicants having gone through that process. So I think that's amazing. Thank you so much for being with us this week. This was a really interesting conversation. And again, Jack and I always joke that by the end of this, a year after doing these podcasts, we're going to have new degrees and biology and
survey knowledge on all these different areas.
Designing surveys, yes. So, Dr. Griffin, again, thank you for being with us. Again, I am Beth Duermeyer. I'm your host of the Idea to Impact podcast, and I also have my co-host,
Jack Manhire.
And we will see you next time.
Bye.